Can SEM be used for biological failure analysis? Imagine peering into the tiny world of cells, tissues, and biological materials with a tool that can reveal details invisible to the naked. That's where scanning electron microscopy, or SEM, comes into play. SEM is a technique that uses a focused beam of electrons to scan the surface of a sample, creating highly detailed images at the nanometer scale. This means it can show a surface features so small that traditional microscopes simply cannot. In biological failure analysis, SEM is a powerful investigative tool. It can examine the surface of medical implants, tissue scaffolds, or biomaterials to find signs of wear, cracks, or corrosion that might cause failure. For example, if a joint replacement fails prematurely, SEM can help identify microscopic cracks or surface degradation that led to the breakdown. It can also reveal contamination or biological deposits that might interfere with the device's function. Another key feature of SEM is its ability to analyze elemental composition through a technique called energy dispersive X-ray spectroscopy, or EDS. This helps determine if there are unwanted materials, such as corrosion products or contaminants, present on the surface. Knowing what elements are involved can point directly to the cause of failure. Using SEM in biological failure analysis is especially helpful because it provides detailed images of fracture surfaces and surface morphology. It can show how tissues or materials have fractured or degraded under stress, revealing patterns like fatigue cracks or brittle fractures. This information helps scientists and engineers understand how and why a failure occurred, so they can improve future designs. For medical devices like stents, prosthetics, or implants, SEM can detect tiny cracks, delamination, or corrosion that might not be visible with other methods. It can also study biological tissues under stress, helping to identify failure mechanisms such as fatigue, contamination, or material incompatibility. In short, SEM is an essential tool in biological failure analysis because it combines high-resolution imaging with elemental analysis. It allows investigators to see surface details and composition at a very small scale, pinpointing the root causes of failure. This helps improve the safety, reliability, and performance of biomedical devices and materials. So yes, SEM can definitely be used for biological failure analysis, providing detailed insights that are crucial for understanding how biological materials and devices break down.